We are the Data Travelers, and this is our pipeline for genomic analysis using DataSpread. Recently, the field of genomics has experienced an explosion of available data due to a rapid decrease in the cost of sequencing with the advent of next-generation sequencing technology at the end of 2007. This basically means that genomic studies can accrue a huge amount of data, much more than would have ever been thought possible, but there are still a lot of problems related to the storage, management, and analysis of this data. In a genome sequencing project, to give a very high-level overview, essentially what will happen is you'll take DNA, randomly fragment it into pieces of different sizes, select a set of fragments that are approximately the same size, and then put them into a library that is analyzed using a platform such as an Illumina machine or a competitor product. Illumina generates short sequencing reads, so it essentially tells you which nucleotides are in your DNA fragment, but only 35 to 150 base pairs of that fragment. Essentially, it grabs onto the beginning and sequences using fluorescence. For many species, there are reference genomes available, which means that these reads can then be mapped onto the reference chromosomes, and that can be done using a number of different programs. In all cases, you'll essentially get the positions of your reads in your reference genome for downstream analysis. Unfortunately, this can be a little bit more complicated. Many studies use a paired end design, where the read isn't just sequenced from the beginning, but from the beginning and the end. So this means that you have a short series of bases at one end and then another short series of bases at the other end, but you don't know anything in between. However, you do know approximately the amount of sequence in between. So this means that the information here is more than just the sum of the parts. Typically, this data is stored in a sequence alignment and mapping format, or SAM file, and that contains the name of the read as well as a flag that identifies the position, whether it's the first or second read in the pair, and also the success of the match. And so this can hold a number of different values and aren't exactly self-evident. There are tools to translate them. This means also that there is no primary key, because the name of the read is the same for the first and second read in the pair, and then the flag is not self-evident. The tuple will also contain the position of in terms of the chromosome that the read mapped to, as well as where on that chromosome, and each tuple also contains the information for the paired read end. These files are often very large, with hundreds of millions of lines or more. These are very useful to genomics because they allow for the study of both mutations and depth in terms of how many reads are mapped into a single area. And researchers may also want to analyze a certain region in depth if they're interested in a certain gene or deletion or something like that. These file formats are ubiquitous across the field of genomics, but the package that's often used to analyze them, which is called SAM tools, has a lot of limitations. It's very slow. It can take 2 or 3 days to sort a file, and it also only performs certain manipulations. So we wanted to capitalize on both the user-friendliness of DataSpread as well as the fact that it has a Postgres backend, which is very well suited to these types of problems where we want to study certain regions or find out if things overlap.
Alright, so now we move to our approach in order to solve the problem. So, number one, we're proposing a tool that will help us import data directly from a SAM file into a bunch of tuples in Postgres. Here we're showing the window to import data. If we try to import a SAM file, DataSpread will say that the type of file is not supported. Now we move to our second proposed solution, that is to create a SAM file toolbox. So that toolbox will have two main functionalities. Number one will be to obtain a primary key for every tuple, interpreting the second column in the SAM file. And number two will be to assist to perform location information queries on the data. Now we move to our completed milestones. So number one, we have DataSpread installed in several of our own servers. It is a really challenging process. Also, we have been capable to upload data into DataSpread successfully. And finally, now we're capable to generate WAR files using IntelliJ IDEA IDE, where WAR stands for Web Application Archive. Now we're seeing DataSpread running in one of our servers. Well, a picture of it, and the idea will be to add the SAM toolbox on top as a plugin.
Getting a SAM file importer to work with DataSpread would go a long way to solving some of the problems that Haley talked about in the beginning. It would make genomic data easier for biologists to work with and give them a familiar tool. So thank you for watching our presentation, and our group is interested in any feedback that you have, so send us a comment.